Dr. Ann Gager, she's Director of Science Communication and Senior Fellow at the Discovery Institute Center for Science and Culture. She received her PhD from the University of Washington and held a postdoctoral fellowship at Harvard University. Her research has focused on two areas: the limits of neo-Darwinism as a mechanism for change, and evidence for the uniqueness of human origins. Her scientific work has been published in Nature, Development, Journal of Biological Chemistry, and Biocomplexity, and she has co-authored the book Science and Human Origins. So it's with pleasure to have Dr. Ann Gager with us this evening.
I'm happy to be with you. I stand before you as a scientist. So one of that class of hopefully rebel scientists who has a different point of view. I'm going to be talking to you about human uniqueness. And I'd like to start off by first of all saying I'm a scientist, but I'm not a materialist. So what I'm going to tell you on the surface may look like I just account for us in terms of molecules, matter, brains, etc. No, I believe that we're more than that. All right, what am I going to talk about specifically? I'm going to tell you about the differences between us and chimps. Why do I choose chimps? Because we don't know what our putative common ancestor looked like. Okay, we're supposed to have descended from a common ancestor, but we don't know what that looks like. I'm going to talk about how much time it would take for us to have evolved. And then I'm going to talk about our origins, and this is where it ties directly into the other talks we've heard. Namely, what are the explanations for where we came from? What are the available explanations? Gradual evolution over time by common ancestry. Guided gradual evolution over time by common ancestry. Well, special creation from the egg of a chimp, or a chimp-like ancestor, or special creation. I'm going to go through these really fast. These are some of the ways we are unique, that distinguish us from chimps. Music, science, theoretical physics, extreme charity. We play chess. We have humor. And we have this silly game where these men line up and face each other and they run this ball down, then they kick it through some uprights. I don't know of any chimps that do that. And most of all, we think about where we came from and they don't. Language distinguishes us from primates. Human language appears to be a unique phenomenon without significant analog in the animal world. There's no reason to suppose the gaps are bridgeable. This is Noam Chomsky speaking. He knows what he's talking about. If you've heard about Nim Chimpsky, any of you heard of Nim Chimpsky? Even his trainers agreed that he didn't speak language, sign language. He wasn't carrying on conversations. He was just saying, I want, give me, that sort of thing. Lascaux, one of the early cave paintings in France. What I want you to notice, look at the bottom of the slide. What do you see there? You see some black dots? There are 12 of them. What do you notice next to the black dots? A rectangle, beginning of math. Anybody heard of the Human Genome Project? We sequenced our DNA. Supposedly that was the blueprint from where we came. Here's a schematic, hand-drawn, so don't pay too much close attention to it, of a piece of DNA in the human genome. The green bars that you see represent exons of a gene. Now, a gene is broken up by intervening sequences called introns. Then there are, at the top you see where it says ALU. Those pink-colored squares are segments of DNA which insert in between or in front of genes, and they regulate their expression. TF sites in blue, those are transcription factor binding sites, places where regulatory proteins come and sit down and regulate the expression of the genes. Lines can function as insulator elements, so that if you have a segment of DNA you want to have expressed as a unit, you can insulate it from the surrounding DNA. attachment sites for attaching the DNA and keeping it localized in one spot. Okay, because you have exons 1, 2, and 3, you can splice those together in different combinations. 1 with 3, 1 with 2, 1, 2, and 3 together. That makes 3 different kinds of proteins from one gene. These are some of the differences in our genome. Now, our genome depends on who you ask, 95% similar, 99% similar to chimps. But there are differences. Now, let me ask you this question. If you took a book and you had another copy of it, well, do we have 2? No, we only have 1. And we had a bunch of knots, N-O-Ts, and butts, B-U-Ts, and put them into the book in targeted places, could you change the meaning of the book? Okay. If you target these gene expression regulators into specific places, change the expression of those genes, where they're turned on, where they're turned off, how much they're turned on, how much they're turned off, you can rearrange the features of the creature that's produced. I can take it even further. This is another schematic representing a series of gene regulatory networks. So the pink one at the top has a lot of, the hub gene has a lot of genes connected to it. The hub gene influences the expression of all those genes that are connected to it. So that it means that it affects the blue and the green networks as well. So if I were to tweak that hub gene, would I change a bunch of expression? Yeah, sure, have a large-scale effect. Indeed, if you look at the gene networks in our brain, even though we may be only 1% different in our DNA, the gene expression in our brain is 17% different from chimps in the cortex, 8% in the caudate nucleus, and 4% in the cerebellum. So it doesn't take much to see that small pieces of information inserted judiciously can have large effects. Okay, our brain on the top, chimp on the bottom. What are the results? We have speech, we have abstract thought, we can draw, represent things graphically. We have a theory of mind. Do you know what that means? It means when I look out at all of you sitting there, I know that you're actually thinking about what I'm saying, right? Okay. Aesthetics, sense of beauty, goodness, truth, morality, and religion. All right, now we talk about some of the anatomical differences. On the left top is a human skeleton, bottom left is a chimpanzee, front and back. If you look at the top right, that's a fossil skeleton called Homo erectus, first in the Homo line that we came from. And then bottom right is Lucy. Anybody heard of Lucy? Okay. Now, if you had to evolve from Lucy to Homo erectus, how many changes would you need? Well, they're listed there. You need a new spine, new pelvis, new rib cage, new legs, new toes and feet, skull, jaws, face, etc. A group, Bramble and Lieberman from Harvard University, did a study of how many physical changes would be necessary to evolve the ability to run from Lucy to Homo erectus. They counted something like 16 different changes, coordinated changes. How many mutations do you think it would take to get 16 coordinated mutations? Anybody? Well, more than 16, right? If you have to get 16 coordinated mutations, do we have enough time? A paper by Durrett and Schmidt from Cornell, they calculated how long you'd have to wait to get one mutation from a human lineage. These are the standard assumptions: population of 10,000, mutation rate, generation time. It would take 6 million years to get a specific mutation in a DNA binding site. How long did it take for us supposedly to evolve from chimps? 6 million years. So that's a problem. What about if you need 2 mutations? They calculated that too, 216 million years. And that's when the first mammals appeared. So my caption says it all. There isn't enough time. Okay, now we get to the fun stuff, origins. When, about 2011, I got an email from a philosopher. And this philosopher asked me, how strong is the evidence against Adam? And I said, I don't know. I haven't thought about it. If Darwinism is wrong, who cares? But I'll go look. So it started me on this journey so that I'm standing here in front of you today. I went into the literature, and it showed me some surprising things, and it continues to show me surprising things. You know that the theistic evolutionists and the evolutionists in general say we had to come from a population of, how much? 10,000, right? From 6 million years to the present, 10,000. Never a bottleneck of 2. Well, over the last few months, on Biologos' website, there's been a conversation going on between theistic evolutionists, scientists, not ID-friendly, over whether there could have been a bottleneck of 2. And finally, one of them got tired of arguing and decided he'd do an experiment. On the left, you have what's called a derived allele frequency. I don't want you to worry about the technical details. If you care about them, you can ask me later. But what you have represented there is the data from the 1,000 Genome Project, and it's got a characteristic curve. On the right is what happens if you start with a population bottleneck of 2 in red and let it go for 500,000 years. Do they look similar? Yes. In fact, if you tweak the conditions of that experiment at 500,000 years, you can match the curve pretty exactly. What does that mean? It means that 500,000 years ago or earlier, there could have been a bottleneck of 2. Now, another scientist said, wow, I'm going to go test that myself. And he went and used another equation, another program. He ran it. And guess what result he got? 500,000 years. And he said, okay, wait, now this can't be right. So there's this other thing called trans-species polymorphism. Big name. I'll call it TSP for short. What it means is, theoretically, if we came by common descent from a common ancestor at the top, and we shared genes, and as time went on, we diverged from one another, so we are represented down at the bottom, orangutans, gorillas, chimps of 2 kinds, and us. Now, can you see how there's some branching going on so that all these species are inheriting some genes in common? That's called trans-species polymorphism. So that would mean that some of these species, say gorilla and chimp and homo, all share the genes in red, called the MHC complex, or HLA, as I refer to it, stands for human leukocyte antigen. It's part of the immune system. Present in all these species. Is it by common descent, or is there another explanation? Could there be another explanation? Sure. What if there's selection driving all of these genes to have similar sequences? Then they would look alike too. Here's what he found out. He used a program that identifies where there is convergent evolution in the genome. He found out that the similarity between these genes appears to be due to convergent evolution. It was such a striking signal that he said, "It's stunning. It's amazing." I asked him about it. He said, "I'm surprised." To sum it all up, it's very simple. A bottleneck of two or a first pair that's older than 500,000 years is possible. Does that mean it had to be 500,000 years ago? Not necessarily. My group is working on a model that doesn't involve evolutionary assumptions, and we are going to be testing different scenarios and seeing if we can demonstrate there could have been a bottleneck or a first pair at a younger age. This is based on the analysis run by two non-ID scientists. It's not me saying it, it's them. Future models may change it, and TSP verifies it. This whole debate has been a surprise to many people. So, to summarize, we are more than our bodies, we are more than our brains, we are more than our genetic code, we are much more than souped-up apes. We could have come from two. Thank you very much.
Biola University prepares Christians to think biblically about everything, from science to business to education and the arts. Learn more at biola.edu.